the next speaker, who I'm pleased to introduce, Osnat Weissberg. And it's nice to see you again, Osnat. We just spent a couple weeks together, along with Daniel Sher and many other great researchers. Doing something that Osnat will talk about the workshop. Where to talk about Alteromonas is because it is, Steve Miller showed, a close associate with marine cyanobacteria. So I will pass it on to Osnat to let us know what she'd like to share with us.
Hi, everyone. Hold on, let me take this away. So I want to tell you about this curation collaborative workshop that we just had a few weeks ago, where we curated a bioinformatics database for Alteromonas that we just heard about from Steve. And this is basically a cyanobacteria friend form, and I'll talk a little bit about that. So why are we talking about Alteromonas? It's the marine heterotroph. It lives in the ocean, but it's not a cyanobacteria. Why are we talking about it in CyanoCyc? Because it's been established as a model organism for interaction with Prochlorococcus, and there's been multiple studies for multiple groups on the interaction between Prochlorococcus and Alteromonas. There seem to be both positive interaction. It helps Prochlorococcus survive in the dark. It helps it survive starvation. It helps it survive multiple stresses. But there's also some inhibition that is going on. So we are seeing both of these. And it is interesting to understand the metabolism of Alteromonas so that we can do a much better understanding of the interaction between the two. And that was basically my as a Prochlorococcus, studying Prochlorococcus, that was my motivation for this workshop. So a little bit about Alteromonas. It's very versatile. It can degrade multiple organic matters, like it can draw down a lot of the carbon in the ocean. Like, as I said, it has both positive and negative interaction with Prochlorococcus. And there's a lot of horizontal gene transfer, so exchanging of organic matter, genetic matters between the different strains. So we wanted to have a much better metabolic picture of the capabilities of Alteromonas, and for that we did an Alteromonas curate-a-thon. I think this is the first collaborative curation effort in bioscience. And what did we do? We wanted to curate a bioscience database for Alteromonas type strains so that as a community researching Alteromonas, we will have good resource for Alteromonas. There were 4 days in the University of Haifa a few weeks ago. We had 20 different participants for 12 research institutes. Most of them were from Israel, but also from the US, Germany, Austria. And these are the PIs that were involved. So just a few pictures to show you that we had quite a good fun on the workshop, but also we, I think, did accomplish quite a lot. What are we trying to do? So if you look at biopsych, tier 1 is something like E. coli. It's been highly curated for 20 years or so. Tier 3 are computationally derived databases. So basically, we took a genome, we threw it into the biopsych backend, and we got a database without any human intervention. And tier 2 are somewhere in the middle. We did some curation, not at the level of E. coli, but good enough to be better quality than the automatic tools. And basically, we thought in this workshop, we did 3 things. One is we wanted to get an encyclopedia. So take all of the existing publication for Alteromonas, or most of them that are related to metabolism, and annotate them on the genome. So attach them to specific proteins, to specific pathways, to specific reactions. Second thing we wanted to do is we wanted to create a high-quality metabolic map. So BioCyc already created some metabolic map, but we wanted to add pathways that were missed, remove pathways that were incorrectly identified, and fill holes in the pathways. And the last thing is build a FBA model that's basically a way to take the genome, put it into a computer, and then you can run experiments in silicon and see what happens in different conditions. That would be the next step. We didn't get that far. How did we approach it? So first of all, we identify gaps and questions as a group. Then we decide together what to work on. Then we split into teams. Each team took a question or a pathway to work on. And we were greatly supported by the curation leader, that was Lisa. And as a result of that, each team came up with a curated BioCyc database, a log, basically a spreadsheet with everything that we did, and text and figures, which we then used to discuss the results and identify things to work on the next day. In addition, we are planning to write a collaborative paper explaining both the process that we went through, but also the pathways we identified and the things that we learned about Alteromonas. Some areas of focus for the workshop. So first of all, we did things that are easy to get under the light. So gap filling, that's basically a list of gaps that BioCyc produces that are enzymes that should be in the organism but couldn't be found. Looking for those, and also curating available literature. So adding notations of this is something, here's a knockdown experiment for this gene, this is what the results, things like that. Then we went into specific pathways. So we looked at carbon metabolism, specifically polysaccharide degradation, C1 metabolism, and mixed acid fermentation. We look at nitrogen-related degradations, so amino acid degradation, nucleotide degradation, and nitrate assimilation, all in the context of if it is feeding organic matter, what could it be? And we looked at interaction-related traits, siderophores, and catalase. All in all, we updated 36 genes, adding evidence codes, summaries, publication references, study interactions, and pathways. I just want to say, we looked at a lot more than 36. These are the ones that we had good confidence that we are on the right track. What is the curation process? We start by selecting a reaction or a function that we're looking for. Then we look for genes with this function by blasting against reference genes and by looking at existing publications for Alteromonas and for others. Then we validate our finding by reverse blasting against, basically taking the genes that we found and seeing that they are actually blasting for this function, additional publications, protein features, domains, operons, and so on. And then once we have a good confidence, we curate the protein by assigning evidence codes, adding summaries, adding reactions, and pathways. Here is an example. So I just want to give a few examples of things like we done. For example, alginate, pectin, and laminarin. These are all marine polysaccharides that are complex carbohydrates, and often they are released from cyanobacteria. To curate these, first we re-annotated the genome using these two databases. Then we compared to A-McLeod 83. Basically, this Alteromonas strain is the model organism for polysaccharide degradation. And then we did the curation using evidence from existing research that did qPCR, comparative genomics, and transcriptomics. As a result, we got these reconstructed pathways. And what you see here in the color coding are genes that we identified. So the yellow ones were already annotated. We just added publications, references. The green ones, we had to re-annotate, add additional reactions, and add the pathway. And the pink ones, things like transporters where we had lower confidence and we still need to do some work to make sure that we identify them correctly. Another example that's slightly different and kind of trying to show the strengths and caveats of reconstructed pathways. These are amino acid degradation pathways in Alteromonas strains for different amino acids. You can see that some amino acids have multiple pathways. Some amino acids have just one pathway, but is across the board. And for some amino acids, there's almost no Alteromonas can degrade them. So this gives you a very good picture of what are the degradation capabilities of this genome. But, however, if we take a closer look, so this is a pathway collage from CyanoCyc that shows you different amino acid degradation pathways leading to the TCA cycle. And all of these red arrows are enzymes that we couldn't find or that are missing in the genome. And there's an open question, are they truly missing, or maybe they are not annotated correctly? But here is something that basically you need to take all of these bioinformatics stuff with a grain of salt. So we are currently setting up growth experiments to see which of these amino acids can actually be grown, can actually be degraded. And then we will work to identify, the enzymes to close all of these holes. Some take-home messages. So first of all, I want to say this workshop, I was a little bit worried before it, but it was really a new perspective of metabolism, a lot less intimidating than I thought, and it was also a lot of fun. We got new insights of some key pathways, and I will definitely take that into my research. It wasn't easy because a lot of the Alteromonas publications are environmental studies or growth experiments, and it's not easy to assign these to a specific gene or a specific pathway. But we use the power of the group, so having multiple complementary viewpoints helped us come up with an informed decision that takes into account multiple people, multiple, like I said, the power of the group. And I think we got better results than if someone would have done that by themselves. Metacyc and biocycs are both very, very powerful and very useful, but were also a little bit overwhelming. So it takes a while to come up with, to use it correctly. But we also used additional databases. I showed you a few, but we also used a lot of UniProt and SwissProt and human logic to make sure that we are getting the correct results. And then, like in all bioinformatics, all of these results are hypotheses or interpretations. So this is only the first step. Currently, we are working with the biocyc team to finalize the move to T2 and to write the collaborative paper. But we are inviting everyone to join us in this ongoing community curation. And the resulting database is going to be open for all. So it will be in biocyc under subscription, but we're also going to have a copy in our lab for people to use using the pathway tools directly. And I can't stop without this. So these are all of the people involved in the workshop, and especially I want to highlight Lisa and Daniel Sher. Lisa was our biocyc curation expert, and she helped guided all of us. And Daniel was the driving force behind this workshop. He made it all happen. So that's it.
Excellent. Thank you very much, Osnat.